All right, so hello everybody back. We now have all of our speakers, and I'll ask you if you're not speaking to mute yourself because we're getting a little bit of echo for the panel on the clinical sessions. So these were all amazing presentations and frankly amazing ways that all the suite of products that we offer can be used to do really relevant modeling and simulation that then affect clinical trials and clinical interpretations and results. So very happy to be here with all our panelists. And I'm going to say we're going to get questions from the audience. So if you would please submit your questions, and I'm talking to the attendees right now on the Q&A, so that we can answer your questions. And also the panelists feel free to, ask questions between each other. That's perfectly fine. I've been asked to close the session at 3 p.m. to give people just a little bit of a chance of a break before going into the closing session this afternoon. So, and again, a reminder that this session is being recorded, so you may be contacted afterwards if you are participating in the session and asking questions. But don't let that deter you from asking questions. So does anybody have anything to start? Then I'll start with Limei, if I may. You were asked a question at the end of your session about sort of the biochemical basis for the effects that you were seeing, and you had to answer that in a very, very little time. So would you mind expanding a little bit on that?
Biochemical basis. I was basically talking about the quantitative system pharmacology modeling about cardiac fibrosis in terms of heart failure. And my model, let's say, is a multi-scale mechanistic model, and we did have a touch-based a little bit about the biochemistry part, but that part is more, I would say, the pharmacokinetics, and then some signaling pathway related to this, how this, this investigated target exposure have effect on the one I mentioned on the pro resolution, agonist, such as FPR2 agonist. They have those effects over there. And then from the biochemistry, reaction, then we actually, those effects will eventually, have effect on the actual organ level, from tissue level to organ level. So, and then the system level. And then because I didn't really, I know I gave a very brief, just a diagram, but in the model, we do have the pulsatile 4-chamber heart dynamics plus the two circulation system, the systemic circulation and pulmonary circulation system. So that part is actually, if you were thinking, let me think about the percentage. Yeah, consider if, like, let's say ODE, the ODE equations in terms of those signaling pathway in the molecular level, it's much less than those ODE equation and level in the organ level and system level. Yeah. Mm-hmm. I hope that answer part of the question, or maybe the audience can be, can ask a more specific, and I can answer.
Yes, and I don't know what's going on. We're not.
But those biochemistry mainly, like I said, it's related to, more focus on related to the cardiac fibrosis, such as related to those, the mediator level change, the neutrophil and macrophages level change. Yeah.
Mm-hmm. And if I remember right, one of the conclusions was that the start time of the dose did not impact the dynamics.
Yeah, basically very minor, very, very minor. Almost you can just ignore them. It's basically no matter which day you start the treatment, the effect is the same. Yeah.
Okay. Very good. And then anybody else from the panel with a question for Limei? No? Okay. The other, thank you, Limei. The other question I had for Julie, I mean, that was a very interesting project in terms of the viral dynamic modeling, and getting, doing the whole dynamic modeling of the viral load, to then get to incorporating the whole PKPD model, to then get to the doses. Can we ask what stage, what was the impact on the whole project? What stage is it now?
Oh, well, it in December it was granted emergency use in the U.S. So it's out there. I mean, the emergency use is a temporary measure, so we're continuing to work on development for full submissions and things, and anticipate other things we'll be able to use this for. So I did want to clarify.
Yes.
So we used viral dynamics, and that was very insightful. But we also used classic empiric exposure response as well. And I actually think there's a lot of value in thinking about mechanistic models together with more empiric learn from the data models. And they actually fed into each other. You would get insights from one way of looking at the data and then bring them to the other model and back and forth. I actually think you get further by sort of flexing with your approaches like that than to say, I'm only going to do one thing.
Yes. And okay, Lizzle, you had a couple questions during your. Thank you, Julie, for that. You had a couple questions during your presentation that was very interesting about the yo-yo dieting. Now, I saw something that when you showed, for example, the changes in ALT versus the absolute values in ALT, there seemed to be an increase in the change in ALT, but not in the absolute in the same time frame. Would that be because it wasn't great enough, the change, that you would have maybe seen it if it was more significant? Or what is the theory behind that? Because the other ones sort of paralleled each other and made a bit more sense. That one, I couldn't figure out why.
Yeah, it's a great question. And I think that's something that we also have, over that 52-week period, right, that you didn't necessarily end up at exactly the same place as you started, right? There was sort of a trending difference. And, to really address that robustly, I think we need to go back and look at, the individual patient responses. But there is like a little bit, we are, even with sort of a very systematic 4 weeks of weight loss, 4 weeks of weight gain, you don't necessarily, like, all of these processes are happening dynamically. They don't necessarily, and because they are sort of, responding in time, they don't necessarily go right back to where the starting place. So you will have like little bits of changes, that sort of manifest over that year, even in things that really fluctuate on a much shorter time scale. So the ALT is derived from the hepatocellular death, and the hepatocellular death in NAFLD is driven by lipotoxicity. And so essentially, from a mechanistic standpoint, when you have that fat accumulation in hepatocytes, and particularly the saturated fatty acids, those stress the hepatocytes. One of the measures that you can look at in terms of that hepatocyte stress is oxidative stress. Those saturated fatty acids can lead to increases in apoptosis and then subsequently in ALT release. So, you can imagine that in a case where you are changing someone's diet, that you are differentially in these different simulated patients, altering the level of fat accumulation in those hepatocytes, and therefore altering the lipotoxicity and altering the ALT. And then there will be some patients that are more sensitive to those changes, where you have more, and others that are not. In general, I would say that you see the percent changes are larger than the absolute changes, in general. And that has been, it's one of the reasons why, as part of our projects, we are often presenting both the absolute differences and the percent differences as part of the deliverable to a client, because you do have to make a decision about how are you going to evaluate your endpoints, right? And there are places where the one type of measurement will be perhaps more insightful than another.
Thank you. I was also curious how it would compare, and I know you didn't talk about that, and I don't know if that work's done, how it would compare the yo-yo versus a steady decrease or a steady increase in weight.
Right. Yeah. So there was a little bit of that shown early on in the previous posters that had been presented. And so in the one case, the 2019 ACOP poster. So there are different ways in which you can generate virtual patients or simulated patients, right? One way, and this is perhaps representative of what might be happening in people, is to slowly have people gain weight in years, decades, right? Simulation time-wise, compute time-wise, that is costly, but it can be done, right? And it has been done in NAFLD-SIM. And if you do that, then you can look at, if you have sort of monotonic increase in weight, you will see monotonic increase in disease progression in the patients. And then similarly with the weight loss, if it's monotonic weight loss, you will see sort of monotonic changes, but I don't know if the changes will be monotonic, because there are multiple feedback loops, and they are happening on different timescales. And so some of those, you might see sort of unexpected increases or decreases that if you follow it out, maybe, follow your mental model of what should be happening, but in the transient, don't always follow what you're expecting. And we've reported on that as well. Yeah.
Thank you so very much. Our next question is, so kind of a mix, one for Sebastian and one for Sebastian and Julie, because those presentations were part of the same bigger, body of work. So a more detailed question is, where does MK4482 convert to NHC? And is it the depot, the place where the conversion occurs? And then the other one is, what do you see as the biggest challenge, and would be for the two of you to talk in the drug development process that model-informed drug delivery can really optimize and have an impact? So Sebastian?
Yeah, I'll take the first one, and I'll let Julie take the second one. So the conversion from the prodrug to NHC is almost instantaneous. So it happens during the first pass through either the gut or the kidney. There is actually very little amount of MK4482 in the blood after administration. Now, the absorption model has this depot, but the depot is not necessarily associated with a specific organ or location in the body. It's mostly a way to achieve a certain shape of absorption, something that is start slow and increase in speed, and then slow down. So it's that quote-unquote sigmoid absorption that can be achieved in various ways. But that's pretty much the intent of that absorption structure.
Very good. And then for the other one is Julie and/or Sebastian.
So you were kind of asking a very big question of what does model-informed drug development bring to the peer review? Is that correct?
Yes, or maybe apply it to the current work, if that's easier. Yeah.
Well, I guess it just to speak to, I do think it had a big role. I mean, obviously, in the middle of the pandemic, once we decided to partner with Ridgeback and develop this drug, this treatment, speed was the key. But speed means not only going fast, but also making good decisions. And we needed really complete, and we did in with basically about a year, a little over a year, completed phase 2 and phase 3. And I think the model-informed approach is really, you need to maximize what you learn from your data. And I think the modeling really helped us do that and to make good decisions to be able to run a phase 3 trial that was successful and to pick the dose, but also gaining insights and things like that you had to treat within 3 days of the symptom or 5 days of symptom onset to for the drug effect to be meaningful. These were important things that we had to really learn quickly during the trials. And I do think the model-informed piece played a big role in that learning. And we continue to apply the model-based approaches, and I think they really do bring additional insights beyond the sort of traditional statistical analysis that would be applied in a clinical trial.
Thank you very much. I have one more, but I'm going to go to a more general one, and people can jump in. For time-varying clearance parameter in Poppk models, is it real or a mathematical device to handle accumulation in the Norman software? Okay, that's, I don't know if anybody wants to tackle that. Sebastian, maybe, or Maxime?
I can attempt to handle this this question. Time-varying clearance. It depends. It could be. It depends on the study. Depends on the time frame of the study mostly. If you have a week of measurements, applying time-varying clearance is questionable. If you have several months, several years of data, and you have body weights that change, if you're dealing with pediatrics data, where the kids obviously grow over time, you need to take that into account. Not in every case, but in a lot of cases, applying a time-varying covariate effect on clearance is a good thing. Time-varying clearance in the absolute term doesn't necessarily. It's not the only. It's not a way to take into account accumulation. You could have a perfectly stable clearance and still have accumulation of your drug. So introducing time-varying clearance or any kind of time-varying parameter is a way to deal with non-stationarity in your PK, not necessarily specifically in the accumulation.
Okay, I hope that was clear. I have one so I'm getting the older ones. I have one for Julie that seems to be a new question. It is, did you identify a PK driver for viral load reduction?
Yeah, it's AUC. And it comes up empirically as the best driver for the viral load effects. But it also, when we look at PK of the triphosphate in cells, that's clearly also best associated with the AUC. So it's AUC seems to drive the that.
Very good. In terms of, there's some of the questions that were asked during the live presentations that maybe we didn't have enough time. And one, actually for Lizzle, I think you already answered the one about the biochemical explanation. And the other one was if you could comment on any key clinical trials impacted by this observation. And then I have a question about Limay's clinical trial right after that.
I think I actually addressed that in the session, but just very briefly, right? That the idea is that in some clinical trials, the placebo group has seen some 10 to 20% of individuals that have shown improvement in fibrosis endpoints. And, you know, does that 10 to 20% matter? It has mattered in some cases. There have been some clinical trials where, you know, it was just tough to see if the experimental drug was working based on the fact that there was a relatively robust response in the placebo group. So I think this does have the potential to impact at least some of the clinical trial interpretation.
Very good. And then there's again the one that was asked before, if you could explain why greater amounts of collagen cause reductions in the cardiac ejection fraction, in case somebody in the audience missed the original presentation. And you're muted, Limei.
Is that the same question during my presentation about the collagen? Oh,
yes.
Okay,
Yes,
I
yes,
see.
yes.
Yeah, somebody asked about, you know, why, you know, the collagen actually reduce the left ventricle ejection fraction. Yeah, because the, you know, during the, let's say during this recurring, this collagen basically will fill in those, replace those scar, right? And it, you know, caused by the myocardial infarction. And this collagen itself, it's not like that compliant, or that's like a, you know, have the same elastance as the original you know, heart like a heart muscle, and therefore because the you know, the ventricle itself, you will become, let's say, less compliant and then much stiffer. So each time when, you know, when the more collagen actually replace those, the dead, you know, let's say the dead heart the dead cardiomyocyte is actually will further reduce the contractility of the left ventricle, and to a certain point, and therefore reduce each time the ejected volume from left ventricle. And that's the numerator of that ratio, ejection fraction. Therefore, it's, you know, reduce the left ventricle ejection fraction.
And the question that I had, thank you about that, about the clinical trial, is you presented that you ended up having a clinical trial that had multiple endpoints with multiple levels, multiple doses and dosing regimens, because you had QDs and BIDs and everything.
Right.
So how did, and that's a pretty comprehensive kind of study where you have a lot of answers coming out of it all at the same time, but it also takes a lot of thinking into how to weigh which portions you're putting into the study, because, you know, you can dilute your answers if you have too much variety in
right
what you have.
Too many cofactors to
consider. Too many cofactors, too many variables. And I go, well, is it because of this? You know, so how did the modeling influence in the design of the study, and what did you learn? Were you able to use some of it to maybe streamline part of the study?
That's a very good question. And, you know, from those prediction, even those prediction, as I mentioned, it's still very preliminary, right? And then it's actually provide, you know, some of the, you know, impact from this, you know, potential, this investigated target on the scar collagen, and then very like a minimal impact on the ejection fraction and the left ventricle volume. And but let's say the clinical development team decided for, you know, in terms of the treatment effect, it seems, you know, because we only want to mainly focus on the cardiac function, it looks like it's minimal. And so they were using those information, they were decided that maybe this, you know, potential target is not good for treatment, but because they still help the wound healing process. So it could be a very potential drug for prevention, not for people who already develop a heart failure after MI, but for people have the MI but hasn't developed the heart failure yet. So prevent them, right? So that's actually, I think it's more meaningful. Yeah.
So, and a little bit back to the impact question, and this should be probably the last set for Pauline and for Maxine and for Sebastian, who had shorter time presenting their posters. If you can expand on how the results of your analysis were applied, or what was the impact of those analyses in the drug development, that'd be very helpful as well. So whoever's ready first.
First.
Okay, so in my case, the work that I've presented in my poster is more of a case study that we use to show as example how to model count data in monoliths. Monoliths can be used very conveniently to model any type of data, so not only continuous, but also count, categorical, event. And so we wanted to model an Alzheimer's disease progression score that have been modeled with a continuous approach, and we applied a count modeling approach from the literature to this data set. There was no following work on this example because it was just a case study, but we hope that it gives a good example for modelers of the field to model their own count data with monoliths.
And then you could, in the future, combine it with information from clinical trials from a drug that is, using the same type of.
Exactly. And we show on the poster how to simulate new clinical trials, also based on the model that have been estimated. So yes.
Yeah. So, Maxime or Sebastian?
I can continue. Yeah. So, just to make a general. When we talk about Poppk model, we do not have real validation criteria like population PK. It's kind of based on the modeler's feeling, if the model has all of the correct mechanisms in there for validations. So we need to build trust in the model. And in this particular project I had for ophthalmic delivery, we are very early currently in the translational aspect. And the goal we have in partnership with the FDA, like the end goal, is basically to have a model that can be used by them and the generic industry to support their generic development. But before we get there, they need to be convinced that the tool we are developing works. And the case I've been presenting is like one of many we are working on. I think now we have around like 12 drugs we've been working on, different formulation, different dose, different rabbits or preclinical species, to demonstrate that we can predict the human exposure in many scenarios and using the tool as so basically what we do, we always use the same strategy to different case studies, and if across all of those case studies we can demonstrate we are successful, then it means that for new drugs for which we do not have the human data, technically the model should be able to predict them. So the case presented by itself is like part of a series, and we are working also on publishing those results. So the community has access to that, and therefore they can use that in their discussion with the FDA to convince also the FDA that the model works, can be used, and they can ask for biowaiver of their clinical studies.
And the more data you have, and as data gets accumulated, you can refine the model and then apply it even better to.
Exactly. It is a game's number, so we need to have new case studies, and when we do the case studies, we discover new mechanisms that we don't have in the model. We put those mechanisms, we repeat everything, etc. So in the end, we have a model that is rock solid and can be used for any drugs.
Very good. And Sebastian, I mean, you had a already sort of an inbuilt story with the presentation from Julie, as well as how your PK model went into the overall analysis. But
I can think about two reasons why the modeling was important in this case. One is technical. The PK data collected in patient was very sparse. There were only a few samples collected in each subject. So using observed data to drive any kind of PD relationship might have been very difficult. Using a model allowed the prediction of overall exposure, and as Julie was mentioning earlier, exposure was most closely related to the effect. So the model allowed that relationship to be a better capture on the PD side. Now, a second example of why modeling was important here. Some of you may know that there are some risk factors associated with the outcome of a COVID infection, especially early on in the pandemic. Heavier subjects, with high body weight, high BMI, tended to have a more severe outcome. And understanding how the body size impact the exposure of molnupiravir was important to know whether there was a need or not to adjust the dose in those patients so they would receive the appropriate exposure and not a subtherapeutic dose. And maybe Julie can expand on that.
I would actually just say, in a very general sense, the Poppk model was the major source of the information that drove the labeling language right now for a wide range of demographic factors, age and gender and all these things. That the data that supports that the same dose can be used in all these individuals really did come from the Poppk.
That's very good. So we see a wide variety of different tools that have been used to really impact the drug development and to learn more from the molecules and develop different types of models that can have different impact on the development of the molecules at different stages as well, from the very beginning to the very end, ending in submission and approval, almost, even if not quite yet. So I don't see any more questions coming in right now. So I want to thank each and every one of our panelists for giving us amazing presentations, very clear, very detailed, and being on hand to answer the questions. And I would ask everybody of the attendees to please attend the closing session, which starts at 3 p.m. today, and whose title is "What Has Changed and Where Are We Headed?" which seems very, very appropriate coming off of this session right now. So thank you very much again, everybody, and you'll get a little bit of a break before the final session. Thanks again.
Thank you.
Thank you. Bye.
Bye.
Bye-bye.
Bye.